Hello, everyone. My name is Gareth Gerrard, and I am the Scientific Director of the Southeast GLH and the Synovus Cancer Genetics Lab at the Southeast Genomics Laboratory Hub based at Guy's Hospital in London in the UK. And Synovus, for those who may not know, is a joint venture between the Guy's and St Thomas's and Trust and King's Health Partners and SynLab. And Synovus provide all of the pathology, including the genomics, which I'm part of, for the Southeast Genomic Medicine Service. And I am here to talk to you today about ctDNA and today's clinical applications. And there are some basic learning aims from this talk. Just a little bit about some of the benefits of liquid biopsies over the more traditional tissue biopsies, the difference between cfDNA and ctDNA, and I will come to these definitions a bit later. Some ctDNA platforms, current uses of ctDNA in clinical diagnostics, and then a bit of focus on the current National Health English Commission services in the UK. And these basically boil down to non-small cell lung cancer and the ER positive, HER2 negative metastatic breast cancer. So liquid biopsies and tissue biopsies. So the standard biopsies generally tend to be a sampling of the actual tumor cells themselves, usually from some sort of needle biopsy. This could be a fine needle biopsy or an EBUS. Of course, in things like colorectal cancers, these could be resection samples as well. But the one thing they all have in common is they're usually quite invasive. There can be some risks associated with sampling of the tissue, and pain, and there can be expense and difficulties associated with, having to get operating theatres, etc., etc. Whereas a liquid biopsy, and there are many liquid biopsies, generally speaking, are much easier to take, quicker. You get a more comprehensive tissue profile because it's not just a single sampling point. It's minimally invasive with reduced risk and pain for the patient. Benefits are things regarding to accessibility as well. For some of these purposes, for the traditional tissue biopsy, the patient may have to travel in to a clinical centre, and sometimes, depending on how invasive it is, they may have to stay over, they may have to have a carer with them. Whereas the benefit of liquid biopsies, particularly those ones which may be based around blood or urine, can be locally obtained, either potentially from home, or it could be, just at a local primary health setting. So some types of liquid biopsy, and there are many. These are just a few examples of the ones we have been using. Blood, which is the source of ctDNA, the circulating tumor DNA, but also liquid cytology. Again, these can be fine needle aspirates, but the main difference here between the more traditional routes is that these liquid tissue biopsies are not typically, or can be, processed in a way which avoid formalin fixation. Ascites and pleural effusions, and CNS, from the central nervous system, but also urine, and you can also argue that saliva could also be sources of liquid biopsy. The main thing all these have in common, maybe with the exception of the cerebral spinal fluid or the central nervous system fluid, is that it's minimally invasive. And these two in particular have potential for long read sequencing, really because you're able to get good quality DNA and RNA out, because these can be processed in formalin-free routes, and it's quite often the formalin fixation, which causes difficulties with fragmentation of nucleic acid. So a little distinction between circulating cell-free DNA, the cfDNA, and circulating tumor DNA, ctDNA. And ctDNA is that proportion of the total free circulating nucleic acid, which is derived from the tumor, because the majority of the circulating DNA particularly in the peripheral blood, will be from non-tumor sources. Because this is short-lived DNA fragments and other nucleic acid fragments, which are released from cells through apoptosis, necrosis, and there are some processes whereby they're also secreted. You get other nucleic acid sources in episomes which are secreted as well from cells. But really for the ctDNA setting, we're thinking about tumor cells. Tumor cells can have quite a high turnover, so you have an increased amount of the DNA fragments being released into the periphery, which can then be sampled through a simple blood sample, and then as long as it is appropriately processed, we can then use that DNA in our downstream diagnostics. Some of the potential uses of ctDNA, and there are many. This is sort of breaking it down into 4 basic things. So there's been a lot of talk about the cancer detection, screening, early diagnosis, but as we all know, there's lots of problems, particularly with the population level screening, particularly of where the majority of people are likely to not be affected, because you tend to have, no matter how good your assay is, you tend to have more false positives than true positives. So there's the potential for harm. But with the introduction of other concepts such as polygenic risk scores into healthcare systems, which is already established in some areas and is becoming more established, then we may then be able to use this technology in already established at-risk groups. And there are also other things, people, for example, who are BRCA carriers or Lynch syndrome families. The main use at the moment is the second one, the molecular profiling and or prognostication, the theranostic application. So this is sort of this is where we're thinking where it's replacing the traditional tumor testing that we have all been establishing over the last decade or so. So this is where you almost have a straight swap of your FFPE tissue, which will be going into most people now use an NGS pathway of some way for lung, breast, colon cancer, etc., etc., etc. And there's lots of advantages associated with doing this. Another use has been around for a while. It's been in research and it's sort of coming into clinical use. Detection of residual disease, particularly following resection. And if you think about the colon cancer setting, where someone may then have a hemicolectomy, and you then look and take a ctDNA sample following that resection, and there's some very good data to show that those who are, there's no ctDNA fraction detectable will do a lot better than those who have a ctDNA fraction, because that's an indicator really that there is some residual tumor. It's already become metastatic in a site or multiple sites, which may be still too small to be properly visualized through MRI or CT. And then going forward, once we've established this, the minimal residual disease sort of setting, monitoring response. So this could be serial liquid biopsy samples to follow people on either targeted therapies or whatever therapy, looking for the occurrence of resistance or any other recurrence, basically. And this usually has a time advantage because you'll often see, there's good data to show that you can see recurrence starting to occur at the ctDNA level before this is visual. That you can see this through MRI or CT tomography. But in this talk, I'm really going to sort of concentrate more on this area because this is more about sort of practical application of what's happening now, and particularly in the NHS in the UK. So some of the benefits of ctDNA over tissue, we've already touched on some of these, but so here we have the human tissues here, and this is the liquid biopsy, really sort of focusing on ctDNA. There are some advantages to having the tissue, and there are many disadvantages, but the advantages really of the liquid biopsy, and one of the things that's really, really good, we've already touched on it being minimally invasive, these low risk of complications. It can capture tumor heterogeneity, both spatial and temporal, particularly for those patients who may be presenting the metastatic, or you were following someone in the metastatic state. They may have multiple metastatic sites, all of which are sort of following a slightly different clonal evolution. So they may have different truncal and branch tumors which are driving them. So if you're only sort of sampling a single site, you may be getting you might find something which is actionable in that particular site, which may not be actionable in one of the other metastatic sites, and vice versa. So you're not getting a complete global picture of the tumor genomic heterogeneity. Liquid biopsy can also be a lot quicker because you can take the blood sample, it can be sent to the lab, and they can just get on and start with the sequencing. Whereas with FFPE and other sort of tissue like that, it needs to go to pathology, it needs to be cut up, it needs to be assessed, it needs to be cut into scrolls, and then you do DNA extraction. So there can be quite a delay, basically. And we know from some of the data we've collected in the lung setting that we know that the liquid biopsy result can be back, sort of before the tissue has even got to the end of the pathology procedure. So there's a lot of advantages with speed, but then there's some disadvantages as well, because there are just some tumors which do not shed as much into the periphery. So there are certain tumor types where the utility of liquid biopsy and ctDNA will be reduced over others, but they may still be there because a lot of these things, it's not going to close down the tumor tissue, the FFPE pathway. We're sort of thinking about this sort of tumor, this plasma first, or ctDNA first approach, where you still have the tissue there as a backup, basically. But what it does mean, if you do get a result you need from the liquid biopsy, it means you don't need to take the tissue any further. So there's actually, from healthcare economic modeling, healthcare economic savings to be leveraged from this plasma first approach, and then having the tissue there as essentially your backup, you can fall back onto. So patients are not going to lose out if, for whatever reason, the liquid biopsy is not giving you the result you need, or is inaccessible for one region or another. And of course, the tissue pathways will still need to exist in many cases, because that is where the actual diagnostic, particularly if someone's at first presentation, then that sampling of the primary tumor will be needed there for proper diagnosis, particularly in sort of things like lung cancer pathways, when you need to properly establish whether it is small cell or non-small cell, whether it is squamous or an adenocarcinoma, etc., etc. So not all blood tubes are the same when it comes to ctDNA and liquid biopsies. I'm speaking to you from England, so of course I'm drinking tea. The traditional EDTA blood tubes, they can be used, but they don't stabilize the blood draw. You get white cell breakdown, and of course then you get release of DNA, which can swamp your tumor DNA. The way to get around this, the EDTA needs to be processed within 4 hours. Indeed, there's lots of data to show that really should be processed within 2 hours. That's kind of doable if it's all being done on a small scale in a single site, but for something that we're running, where we're a genomic laboratory hub, and we operate on a sort of hub and spoke model, and we are having samples sent from all over quite a large region. That's not going to work for us. Luckily, there are some very well established ctDNA stabilization tube technology we can draw upon. And what this does then stabilizes the white blood cells, which means that they then don't break down, they don't release their DNA and other lytic materials, which means that the ctDNA component is not going to be swamped by all this sort of germline white cell DNA. So we will not, in our center, we will not accept EDTA tubes at all. We will only accept one of the already established ctDNA stabilization tubes, such as those available from Strec or QIAGEN vaccine tubes, etc. So a typical ctDNA extraction workflow, again, you start with your blood collection in your ctDNA stabilization tube of choice, or what has been mandated by your local genomic center. The plasma preparation, because what we really want to do is we want to get rid of all the red cell components and the white cell component. So that white cell component can be useful in some applications, but really the ctDNA is living here in the plasma component, and it's a two-spin protocol. You have a sort of lower speed spin to get the separation of the plasma, the buffy coats and the red cells. You then take off this plasma component very carefully, making sure you don't disturb the buffy coat, into a smaller high spin tube, and then you spin a much higher then, and that's just to get all the sort of cellular fragments down here and the ctDNA still floating here, because it's so small, it's not affected by gravity in quite the same way as cell fragments. Take that through then to ctDNA extraction or cfDNA extraction. There's various ways of doing this, and there's plenty of platforms out there. They're either going to be membrane-based or bead-based of one form or another, though there are some innovative techniques coming into this space. And then this can then go into downstream applications. You do your QC and quant first to make sure you've actually got some ctDNA there. You can run it out on a tape station or something equivalent to look for that characteristic. And so ctDNA is, all cfDNA is fragmented into that sort of 140, 160 base pairs sort of range. So if you run this out on some sort of capillary electrophoresis, such as the tape station, you're going to see a peak in that sort of range. Downstream applications, NGS, PCR, digital PCR, etc., etc., etc. And then the bioinformatic pathways for you to do your data analysis. And then, if it's for clinical purposes, you're going to be linking this out to clinical grade decision support software, linking into all the, guidelines and clinical trial databases. So methods of ctDNA analysis. I mean, again, this is very reductive. There's always innovative ways of looking at these things. I mean, and again, what we're interested in, the limit of detections and the number of variants detected and the throughput as well, because what we're doing now in the UK, we're really thinking about industrializing a lot of these processes, because we're the hubs for quite large populations of people. Real-time PCR, very well established. Again, you can only really look at hotspots. You're relying on primer and probe technology. Digital PCR, again, primer probe, hotspot based, but you've got a lower limit of detection. Actually go a bit lower than this. I think digital PCR living a bit, I think you could probably go down a bit more than that. And then NGS, again, I think 0.01 you can in some applications, but most of the sort of the commercial kits which we'll use really go down to 0.1. You could probably swap these two around. So we're really looking in that sort of 0.1 to 1 to 2% range. The advantage, of course, the NGS, you can use panels, you can use Aplicaciones, you can use a hybrid capture pull down. So you can look at lots of different genes, lots of different regions, doesn't have to be hotspot driven. And of course, you can multiplex, so you can look at many patients at the same time. Of course, the difference when you're looking at ctDNA over sort of NGS analysis from FFPE or other sources is you need to sequence much deeper. In order to get that extra limited detection, you're looking at 10 to 20 times the depth. And if you think in a sort of typical somatic panel, you're looking at 600, 700, really up to about 1,000 depth. So really for a good quality ctDNA panel, then you're thinking 10,000 depth, 20,000 reads across certain regions to really get down and get that limited detection that you're looking for. Quick comparison, some of the ctDNA panels, I picked these because these are the ones that we have been familiar with using in our own service. We have sort of in-house and send away. In-house, we originally looked at, we still have this platform from Thermo Fisher, it's the Genexus, and OPA is the Oncomine Precision Assay. We chose this because we use this for lung cancers, and it works from FFPE and from ctDNA. In fact, it's ctDNA, so it's circulating tumor total nucleic acid. So that's a mix of DNA and RNA. With advantage, then you can look at fusions in the RNA. Whereas these other panels are all ctDNA. They do have some fusion calling abilities, and that's because then you've got hybrid capture baits designed across the usually intronic most common breakpoint regions. So it's usually just based around ALK and RET and ROS, for example. The foundation one is a bit more expansive. It goes out into things like some of the NTRX and FGFR2, etc. Which is very handy. But of course, we tend to use this more for the solid tumours, and really it's not a huge number of, particularly the adult solid tumours outside of, if you take the sarcomas and brain tumours out and the hematological tumours, then the adult solid tumours, there's not that many of them which are fusion driven, cholangiocarcinomas and even lung cancers, it's the minority of them. But you do want to catch them when you get them there, particularly the ALK and the ROS and RET, because we have some good inhibitors for those. NTRK, yeah, there's really good inhibitors, but they're incredibly rare. Just, I don't know, we see like 1 or 2 a year in our service, and we have a big service. So number of genes, Thermo Fisher, the OPA, it's quite a small panel. This is a medium-sized panel, about 50 genes. The Sophia Genetic MSK Access, so this is the Memorial Sloan Kettering panel that was developed there, and Sophia have commercialized this and sort of made some changes to try and improve it. We also have this in-house. We've also used these send-away assays from Guardant, the Guardant 360 and the Roche Foundation. So MSK Access, 146 genes, useful, good pan-tumor panel. Guardant 360 CDX, 74 genes. It's pretty good. Slightly a bit bigger than the Genexus, but it's a pan-tumor panel. You probably want a few more genes in there, and the Roche Foundation one is 324 genes. So that's, you've got a lot being called in there. Yeah, I mean, these sort of things come then with higher then data burden, higher bioinformatic burden, a higher sort of interpretation burden. The ctDNA, whether it can do RNA as well, it's only the Thermo Fisher Genexus, so that can call fusions from RNA. Very difficult to validate that because there's no commercial control for that. We did our best. Chemistry, Amplicon and the rest of them are hybrid capture. Can they do CNVs? Yeah, they can all do CNVs. Copy number gains, definitely. Copy number losses are a little bit more tricky, but we have seen some particularly things like P10 losses being called. MSI, microsatellite instability, particularly useful for those tumors which might be eligible for checkpoint inhibitors and standard care, and also, of course, Lynch syndrome marker. They can all do this apart from the Thermo Fisher. It's just they just haven't painted across that. They haven't got the design for it. Again, mutation burden, TMB, the Thermo Fisher OPA can't, just too small, but the other three can. I think in the Sophia Genetics one and the Guardant one, it's there, it's available, but it's kind of not validated. It's there sort of for information purposes only, whereas the foundation one really does have that. This is the clonal hematopoiesis of indeterminate potential, and we'll come on to a little bit more about this. But this is essentially white blood cells which have got themselves some clones which may or may not burn out, may be associated with myeloproliferative disorder or lymphoproliferative disorder. Most of the time, it's just a sort of false positive. The OPA, none of these can deal with apart from they can. The Sophia Genetics does this through simultaneously sequencing some of the germline from the buffy coat, and then you can do a subtraction, so it knows what's coming from the white cells and what's coming from the tumor. The Guardant and the Roche, what they tend to do is they just flag these things as being potentially CHIP, because there's a lot of the well-known genes associated with hematopoiesis, DMT3A, TET2, ASLX1, but you do see some tumor-associated, ATM tends to come up a lot, and there's some data to show that things that you can get this CHIP signal giving you sort of BRCA2 variants, which obviously, if you're going to be looking at things like prostate patients and maybe even ovarian or breast patients, then you don't want to have BRCA coming up in your CHIP signal, because that's going to be a disaster. So the real advantage over that Sophia Genetics is that it does the germline. You can subtract the chip signal, and also it means you can then ratify and validate findings that look like germline, which in the UK now we're starting to flag these and say this looks like it's germline. It means that you've already done your test, basically, and it just need to be looked at by clinical genetics colleagues to ascertain its pathogenicity. Regulation CIVD. Most things still haven't quite come into the IVDR range yet, particularly not in the UK, because we're still in this sort of regulatory black hole a bit, since we are sadly no longer part of the European Union. But CIVD is still recognised. So there is a CIVD version of the OPA assay, but we don't use it. I think the MSK assay, they are looking to IVDR parts of this, but both the Guardant and the Roche are CIVD, so they are validated for clinical use. So, great question. For clinical diagnostics, is ctDNA analysis as good as tissue? This is some real-world data coming from colleagues in North Thames at Marsden and the ICR as part of their sort of pilot for this. And they show that actually in the lung cancer setting, I think this is where this data is coming from, and it's showing the ctDNA variants here, and these are the tissue variants. These are the ones that were common to both of them, and these are the ones which are unique, that were only just detected on the tissue, only detected in ctDNA, and these are all tier 1 variants that would have been loaded by the variants as well. These are the ones which are clinically actionable, and actually you can see that ctDNA seems to show that it has a bit of an advantage. So okay, that's pretty good. And as we know, there's some tumours don't shed as well, so there will be a proportion of your ctDNA samples which are there's no information, basically. You can't make a call on it, particularly if you're using an assay which doesn't give you a estimation of tumor fraction, which not all of them do. In fact, actually, I think it's only really the foundation that does at the moment. So you might see something in there, but you don't really know whether it's actually coming from the tumor or not. That's good. There's no, there seems to be a small advantage there to the ctDNA. And of course, the other great benefit is the turnaround times. So we have the standard of care tissue here, and you can see there's some outliers here because this is real world data. These are obviously samples with a different issue for one reason or another. And here's the ctDNA component, and you can see here median of 8 days versus 22 days. So that's a great advantage, and particularly in the lung setting. Some of these patients will have ALK fusions, and we already know that every day counts, basically. And of course, this getting patients onto targeted therapies or into appropriate therapy sooner leads to better healthcare outcomes, which means that you're not having to do quite so many invasive procedures quite so early on. And it's better for the patient because they've had a less invasive test and they're on to the appropriate therapy quicker. They're not hanging around full of anxiety, waiting to hear what the results of their test was. Because obviously, if you already have lung cancer, that's bad. But if you find out that you've got an EGFR-driven one, that's good because you can immediately go on a very effective therapy straight away. So that's fine. Okay, you've got a KRAS. Well, at least you can go on to hopefully some immunotherapy. That will it'll be beneficial. So this brings into this plasma first approach, particularly for non-small cell lung cancer. Probably sort of makes sense in other tumors as well. Really for the non-small lung cancer, because a lot of the molecular diagnostics we do for the other tumors tend to be the other end, tend to be when they've gone metastatic, or when they have failed on first line therapy. Think being colorectal cancer. That's often there's stage 1, 2, 3A, whatever. Then it's hemicolectomy, and for a lot of patients that tend to the story, it's only when they go metastatic you need to see whether they've got KRAS or NRAS or BRAF. For the lung cancer, obviously you need to know straight up. You want to know you want to molecularly characterize that tumor to see if it's EGFR driven or ALK driven or ROS driven or whatever the situation is, so that you can put them onto this targeted therapy. For the tissue, that's what we've always got. That's always going to be there for the lung cancer. Still need to diagnose. Still need a pathologist looking at a microscope, looking at those cells. Is it adenocarcinoma? Is it a small cell? And then doing IHC stains to see whether it's squamous or whatever. But the overall healthcare savings means there's still a healthcare economic benefit going for this blood draw first. Again, we can see that you can do this blood draw at radiological suspicion. And by the time you get the results back, it can be back before patient has even been booked in for an EBUS, or it's still being processed. It's not even been sent to the genomic lab yet for follow-up. Then that's still there if the ctDNA analysis was uninformative. The tumor is too small. It's just not shedding enough. It's one of those solid tumors. It's not so well vascularized, or it's got a lower apoptotic index, which means that it's just not shedding and it's not detectable. We've always got the tissue there, but it also means that if this is informative and you see an EGFR exon 19 deletion or whatever, it means that you don't need to send the tissue block. It's going to stay with pathology. It doesn't need to be sent through to genomics, which means that there is a saving, which means also there's more tissue then left for those tricky cases where you need to run some more IHC screens to make sure that it is what the pathologist thinks it is. Clonal hematopoiesis, CH, sometimes CHIP, and clonal hematopoiesis of indeterminate potential. So this is the white cell signal. They can sometimes get in the way. There's a lot of it's associated with age. We just start accumulating these slightly dodgy white cell clones just because we've been knocking around for a while. A lot of these can give you, they're associated with sort of hematopoietic genes, sort of things you'll see in, MDS and myeloproliferative neoplasms. Some of these patients, because the greatest risk factor for cancer is age. So a lot of our cancer patients will be old anyway, or older compared to the population. Some of these will have actual legit, hematopoietic stuff, but it means there's a potential for catching some of these things earlier. So exactly that. But it is really important to make sure that we know this is definitely not coming from the solid tumor we're looking at. Like I say, you'll see TP53, you'll see ATM, sometimes you'll see BRCA. So all of these things, could then muddy the picture for how we then treat our patients based on liquid biopsy results. And like I say, the advantage of the MSK Access is that it's able to subtract this. It's able to, it runs some of that white cell DNA as well, and alongside the ctDNA, so we're able to determine what is from the tumor and what is from, the white cells that are also in your blood draw. So a little bit about the genomic structure in the UK, specifically in England. I know this is an international audience, so the United Kingdom is made up of England, Scotland, Wales, and Northern Ireland. In England, we have the NHS England, and then similar bodies in Wales and Scotland and Northern Ireland. But in England, which has got the largest population, all genomic testing has been centralized into these 7 genomic laboratory hubs. And Synovis and I work in the southeast here, so covering South London, Kent, and various Surrey, Sussex. Some coverage of about 8 million people, 8.5 billion people, I think it is now. That's quite a large population, Scandinavian country or two. So we have to think about how we do these things at scale. And we've seen particularly our cancer service increase by about 500% in the last 4 years, and it's looking to sort of half again, in the next 6 months or so, particularly with the onboarding of ctDNA. In order to get ctDNA technology into the clinical space, into the NHS, the National Health Service in the UK, NHS England being specifically for England, there was a pilot scheme which was funded and undertaken across the 7 GLH regions. It was actually run by what was called the GMSA. This is the Genomic Medicine Service Alliance, which is all the sort of hospital trust infrastructure that was needed in order to coordinate and allow the actual genomic laboratory hubs to do the work. It was 3 phases, and all the sequencing was done at just 2 centres. Most of the work was done in the North Thames at the Royal Marsden Hospital, and then some also was done in the Northwest. Though it says here Northeast because the coordination was coming out of that, because Alistair Greystoke was the PI for this, and had been running similar sort of translation research schemes into this area to really start that initial data gathering. We ran quite a lot of samples. There was somewhere in the region of 12,000 samples run across the regions. And there was also then not just a technical analysis, there was also a healthcare economic analysis that was going on as well. We were able to present some of the data from our own region, looking at this. Again, looking at these real-world insights into the data that we gathered as part of this pilot. We can see here that we got, we again saw that decrease in turnaround time, which we were looking for. And also we saw that there was really good concordance between the tissue and the liquid biopsy, again with a little bit of a sort of end to the liquid biopsy. Moving into ctDNA analysis of breast cancer. So lung cancer became established, and then for breast cancer. And really the push for the ctDNA really came for this resistance to aromatase inhibitors in the ER positive, HER2 negative setting, metastatic breast cancer for patients progressing on aromatase inhibitors, etc. But also there's use then for PIK3CA, and then now we also have AKT and PTEN for these PI3 kinase pathway inhibitors. So ctDNA for ESR1 in ER-positive, HER2-negative breast cancer, and these are acquired variants that develop as a response of endocrine therapy, such as aromatase inhibitors, and typically following first or second line of therapy. And so there was a new class of the selective estrogen receptor degraders, or third therapy, and the first of these was going through clinical evaluation through NICE, which is our accreditation, clinical accreditation scheme in the UK. So we were all asked then to bring on our ctDNA technology. This is how it works. They're activating variants in the gene that encodes for the estrogen receptor. And what it means, they then start signaling in an estrogen independent manner. So when you're using, endocrine therapy, aromatase inhibitors, that stops working because it becomes estrogen, independent essentially. Again, some very well characterized hotspot mutations, variants in the ESR1 gene. And the whole thing about it really benefit the ctDNA is this could be polyclonal, because there's so many of these, and of course you can have multiple metastatic sites and they can all have different variants. So just sampling a single site might not give you the answer you need, and you might want to do this sequentially as well to look for development of other resistance variants. So it's ctDNA is recommended for this. Alesterant is the first SERD. It's already available. Third is available through the NHS, and this is a nationally commissioned service now in the UK. Because we're also bringing this technology up for ESR1, the other genes of interest in breast cancer, PIK3CA, but then also AKT and PTEN now because of the new AKT inhibitors, it made sense to do these by ctDNA as well if we're already be doing the ESR1, because it's basically the same patient cohort. There is some changes in the eligibility because these tend to be sort of early driver variants, whereas the ESR1 variants, like they're acquired. And this is really because we have alpelisib for PIK3CA that's been established around for a while, but just really come into NICE sort of commission service in the UK, the AKT inhibitor cabozantinib, which again this required us not to look at PIK3CA but also PTEN and AKT alterations. And this is just a little bit of our validation data for this service, looking at the scope of the variants we were picking up. Because again, we weren't just looking at one, though we were looking at the whole thing and reporting everything. So ESR1, and PIK3CA, because they can co-occur. They're not different, they're different, completely different pathways. PIK3CA by itself. You can say the AKT1 or PTEN, they're actually a very small number. These others that include TP53 and other variants. Actually, very small failure rate, about 2%. And for the majority of samples, you don't actually pick anything up at all. So that is a whistle-stop guide to current clinical applications, particularly in the UK and the NHS, particularly NHS England. And these are some of the key people. The main person with it is Persephone Park. This has really been her project. She's the one who's driven this. It's been her energy and who's really made this happen in our centre. But also Alistair, Sasha, Rochelle, everyone else here. Nirupa, Nicola was the head of clinical genetics, who sort of always supported us and let us get on with this. And Sammy Richard Hall, my co-director, who runs the operations side of things, and James Fisher, who's really sort of like gone out to bat with us, the people who need to unlock the money at Synovus. So thank you very much for your attention.